So I am very happy to have the opportunity to give this address in front of an audience of paleontologists. So I'm not a paleontologist at all. I'm an evolutionary biologist at the molecular level. But I think we have many questions and results to discuss and to exchange. And that will be the topic of this talk. So I want to introduce molecular thermometers, so which is a way to use extant molecules to estimate ancestral temperatures. So you know the current view of the tree of life based on molecules. So this view identifies three primary domains in the extant life. So the two prokaryotic domains, bacteria and archaea, and the eukaryotic domain. So the last common ancestor shared by all these organisms is now often and conveniently called LUCA, for last universal common ancestor. So a feature that was rapidly noted when looking at this view of the universal tree is that the location of taxa containing thermophilic or hyperthermophilic organisms. So as you can see, those taxa tend to be located in places of the tree that are apparently early emerging. And so this simple observation gave rise to a hypothesis that became very popular. Excuse me. That became very popular, and that the last universal common ancestor was itself a thermophilic organism by a parsimonious argument that if the deep branches of the tree are thermophilic, then their last common ancestor should have been probably itself thermophilic. So what we try to do is to devise an empirical test of this hypothesis that became very, very common, and to base that on empirical data, which are molecular sequences. So in terms of vocabulary, I'll be talking about the optimal growth temperature of a microbial organism, which is the temperature at which this organism grows fastest. And we can conveniently define three categories. For those that grow above 60 degrees are called thermophiles, and those that grow above 80 degrees are usually called hyperthermophiles. So no eukaryote is fancy enough to be able to grow at such high temperatures. So this concept is relevant for prokaryotes, that is for bacteria and for archaea. So the outline of what I'm going to present today. First, what is the link between biological macromolecules, that is between RNA sequences and protein sequences, and temperature. Then how to estimate accurately ancestral temperatures. Then how we apply that to two empirical data sets. And how can we interpret the results of our measurements. So the first part, how is it possible to connect biomolecules and temperatures? So you know the ribosome, a molecular machine that is the basis of the protein synthesis. So it's made up of 2 subunits, and each subunit is an assemblage of large RNA molecules and of proteins. So the large RNA molecules have a very precise 3-dimensional shape, which is crucial for the functioning of this molecular machine, which is made up of a series of regions that are in double helical structure and of loops that connect these double helices. So in a few words, those helices are crucial to the functioning of the ribosome and therefore to the functioning of the organism. So in a more abstract representation of the large of the RNA of the large subunit of the ribosome, we can express it this way, which is called the 2-dimensional, the secondary structure. We see a series of helices connected by loops and helices again, and so on. And so these helices are made of base pairs, complementary base pairs, which are nearly exclusively either GC or AU pairs. And there is a clear chemical difference between these 2 pairs, among others. It's the number of hydrogen bonds involved in each pair of complementary bases. So 3 for a GC pair and 2 for an AU pair. So this very simple fact has a very clear consequence, which is that a molecule to resist to high temperatures need to enrich its ribosomal RNA, needs to enrich its base pairs in G and C to have more hydrogen bonds and so to resist to thermal movement. And this is very clearly supported by the empirical data, where you see for a large number of bacteria and archaeal genera, we see that there is a very strict correlation between the optimum growth temperature of these microbial genera and the GC content of their ribosomal RNAs. And more specifically, this applies to the part of these molecules that is in double-stranded form, what are called stems. Okay. An unexpected feature of molecules is that the correlation between GC content and temperature is specific of structural RNA molecules. It does not apply at the genome scale. So if one considers the GC content of the complete genomes, there is no correlation between this content and the optimal growth temperature of the microbe. So you can be a thermophile and be poor in GC. You can be a mesophile and be rich in GC. Okay. Now, the other large category of large biomolecules are proteins, and it occurs that also protein sequences in microbes are influenced by their temperature. So these are the protein content of organisms at large, that is the global proteome, the fraction of each amino acid observed in all proteins of an organism. So there is a number of ways to represent how these vectors of 20 amino acid frequencies varies across organisms. A very simple way is to count the frequency of these 7 organisms, so isoleucine, valine, tyrosine, and so on, and to plot that at the function of the optimal growth temperature of the organism. And we see again a very clear correlation between amino acid composition of proteins and the temperature at which these organisms grow. Okay? And so this is my molecular thermometer. using these positive correlations between molecular feature and environmental temperature, it is possible, if one can estimate the base or the amino acid composition of an ancestral organism, then through this correlation we can estimate, we can deduce the environmental temperature at which this organism lived. So crucially, to estimate accurately ancestral environmental temperatures, we need to be able to estimate accurately ancestral molecular compositions, that is ancestral GC content, ancestral amino acid compositions of proteins. Okay? And so here there is a technical point that is crucial to the argument, and so that I cannot present you the argument without delving a little bit into this technical argument. So molecular evolution is modeled in a very precise framework now. It is a probabilistic framework that describes the evolution of an ancestral molecule along a tree through a succession of speciations that represents divergent nodes in the tree. So we have nodes that are ancestral species, and we have the extant species at the tips of the tree. The branches of the tree represent evolutionary lineages where molecules vary in sequence with evolutionary time. And the modeling of this process uses a matrix Q, which is the probability for that each amino acid gets transformed into another amino acid along the branch. And so the way the modeling is commonly used is to make the hypothesis to assume that this matrix Q can be decomposed into the product of another matrix that is called the exchangeability matrix and a diagonal matrix of equilibrium frequencies. So this first matrix, denoted S, represents the propensity of each amino acid to be replaced by another one. So you have large dots that represent, for example, this large dot represents that leucine and valine are frequently exchanged in protein sequences. And these are equilibrium frequencies, so these represent the composition of the molecule toward which any molecule evolves when evolutionary time accumulates. And so the crucial element I want to point out here is that the common modeling which assume that the same exchangeabilities and the same equilibrium frequencies apply to all branches, that is, apply all along evolutionary time, as a consequence that there is a unique composition. A unique composition is shared by all molecules under consideration. And this contradicts biological evidence, because I showed you that molecules, extant molecules, differ extensively in terms of composition. Some are GC rich, some are GC poor. And so we need to change this modeling to account for what we observe in extant sequences. And so this is what we did. And we propose to use a more parameter-rich model in which we eliminate the assumption that the vector of equilibrium frequencies is the same shared by all branches. Here we construct a model in which there is a vector of equilibrium frequencies for each branches of the tree. And so we allow one branch to enrich in GC and its sister branch, for example, to deprive in GC a sequence. So our model becomes able to represent the evolution of molecules whose composition varies along time. Okay, and so this is what we did. So there is a lot of technical difficulties to handle such a parameter-rich model. And our data sets are two. One of nucleotide sequences, so the ribosome RNA sequences from a large number of bacteria, archaea, and eukaryotes. And another smaller data set of protein sequences for 30 organisms. So it is a question of computational tractability that had constrained us to work with much smaller number of sequences at the protein level than at the DNA level. And with this model, we've been able to estimate the ancestral compositions of our sequences. So ancestral base composition for RNA sequences, ancestral amino acid composition for protein sequences. So this is what the data look like for those of you who are not familiar with dealing with molecular data. So we have certain number of lines that represent organisms, extant organisms, and a large number of columns, so several thousand such columns that are filled with one or another amino acid observed in extant organisms. So we have used data analysis technique called correspondence analysis that is very similar to principal component analysis to build a measurement of the amino acid composition of proteins. And so this data analysis method constructs the factors by which the data vary most. And we see that the second factor of this analysis, so the second axis of principal component analysis, is very strongly related with environmental temperature. You see that red dots are on one side of this factor, while green dots for mesophytes are on the other side of this axis. So if we draw this second factor of the analysis as a function of optimal growth temperature, we see this clear correlation appear with the linear correlation coefficient of 0.83. With ribosome RNA sequences, this is our molecular thermometer. We have this clear correlation between the GC content of the molecules and the optimal growth temperature with a correlation coefficient of 0.79. So what we did is estimate the ancestral composition at all nodes of those trees, and we are especially interested in some deep nodes in this tree. The last common ancestor of the bacterial domain in blue, in green the last common ancestor of the archaeal domain, and in red their common ancestor to these two domain ancestors, the last universal common ancestor. And so this is done separately on two data sets, so two independent analyses, one ribosome RNA data set and one a protein data set. Since ribosome RNA genes don't encode for proteins, these two data sets are independent from each other. And we see qualitatively the same trend emerge from this analysis. That is, the data support thermophilic or hyperthermophilic domain ancestors. The blue line and the green line in both cases are high temperature, and a clearly lower temperature, the red line, for the last universal cellular ancestor, and with non-overlapping, with statistically significant differences between these estimates. So it is conveniently possible to visualize it with this tree in which colors correspond to estimated environmental temperatures. Blue for cold and until red for very high 90-degree environmental temperature. And the estimates we obtained is about 70 degree for the bacterial domain ancestor. So this is the bacterial domain B. And then here we have the archaeal domain A. And we have about a little bit less than 70 degrees estimation for the temperature at which this domain ancestor lived. And what is very surprising is that we have a non-parsimonious estimation of the temperature at which their common ancestor lived, because we have a low estimate for the temperature at which Luca lived, which is not at all the average temperatures of these bacterial domain and archaeal domain ancestors. Okay. So is it possible to make sense of these, what those molecular thermometers tell us? So the first inference is that the ancestral state of the bacterial domain and the archaeal domain is thermophilic. So other molecular evidence support that. A very different procedure to study this question is to estimate ancestral sequences for a protein coding gene, then synthesize those sequences, then clone those sequences in E. coli, harvest the protein produced by these genetically modified E. coli cells, and measure the temperature at which the protein unfolds. And so this is a large tree that you can't read, but that is the bacterial domain. So a whole lot of different bacteria put in a tree, and this part of the tree is the ancestor of the bacterial domain. And you see that when you go deeper in the tree, the ancestral proteins are more thermostable. Okay. In terms of the geological record, so this is where you know much more than I do. My understanding is that there is isotopic evidence that, interpreted in a certain way, would support a trend going back in time towards warmer and warmer global ocean. My understanding is that the connection between those isotopes and the temperature of the global ocean is somehow put under question, but that you will be able to tell me probably. But let's say if this view is correct, then this would be the same kind of trend as the one we observe with our molecular thermometers. Now, our second inference is that from that very early point in time of the bacterial domain ancestor and the archaeal domain ancestor, if we go even further in time, temperatures would have been lower. So there would be a transition from low to high temperature at some point in time. I think there is support for such kind of events in the geological record, namely about giant asteroid impacts about 3.3 billion years ago that could have heated the atmosphere, boiled the ocean surface, evaporated tens of meters to perhaps 100 meters of seawater. And such events would have severely affected microbial communities and would have profoundly affected biological evolution until 3.2 gigayears ago or even later. So maybe what we see in our molecular thermometers is a reflection of these events. So early forms of life would have evolved before those giant asteroid impacts would have killed all mesophilic forms of life, and only thermophilic forms would have survived, and then would have diversified again and given rise to mesophilic forms of life from that thermophilic ancestral state. So an idea I would like to advocate in front of you is also that maybe this could be an alternative procedure to do something that is notoriously difficult, that is to give date to molecular phylogenetic trees. So maybe we could have a two-step procedure. One first step would use molecular trees to estimate average environmental temperatures at which these molecules were submitted. And then the second step would use the fossil record to reconstruct past temperatures and to match those two temperatures. Maybe that would bring a little help how to put date on those molecular trees that have the very bad habit of not living fossils and so not being able to be dated. How am I with time? It's okay. So just a few more points to present some more recent analysis we did on this topic. So here the intention was to increase significantly the sampling of the archaeal domain. So you see previously there were less than 10 archaeal genomes used. Now we have a much more intense sampling of this domain, and the same conclusion are supported. Thermophilic state for the ancestral of this domain, and also for the ancestral of the bacterial domain, and the mesophilic state for their last common ancestor. And this we can do again on the protein tree I showed before, and on the nucleotide sequence tree I show here. So again, we have the same general feature that emerges. And another feature interesting in terms of trying to date molecular trees is that there is very clearly a relationship between the length of those branches. You see long branches here and short branches there, and their color. So lineages that remained thermophilic tend to be short lineages, whereas lineages that evolve from an ancestral thermophilic state to an extant mesophilic state tend to be quite long. So this is interesting because it indicates and detects what is a major determinant of molecular evolutionary rates in archaea. This major determinant would be environmental temperature, and it is the variation of the molecular evolutionary rates that creates difficulty in dating molecular tree. So better understanding determinants of these evolutionary rates are necessary for progressing in this direction. Finally, I would like to show a few more control experiments trying to understand more the modeling we are doing. So first one, I told you it is very important to use what we call the non-homogeneous evolutionary modeling. That is, not assume that all branches evolve with the same frequency parameters. So I would show you the effect of doing that or not doing that. Then what is the sensitivity to the uncertainties in the tree of life? Because of course there are many uncertainties in the precise shape of the tree of life. And what information makes possible this non-parsimonious result we observed? That is, the ancestor of two thermophilic organisms is not predicted to be itself thermophilic. So the effect of the non-homogeneous model, we see it here. So with the non-homogeneous model, we have very different estimates of the temperatures for the bacterial ancestor, the archaeal ancestor, and for their common ancestor, Luca. Whereas with the homogeneous model, all these temperatures are very close to each other, to one another, because the model bioconstruction is not able to make compositions vary, and therefore not able to make temperature estimates vary. Now, in terms of uncertainty about the tree of life, well, there are plenty, especially when we deal with such a difficult question about so ancient events. So the three-domain hypothesis has been very popular, that life is divided in bacteria, in archaea, and in eukaryotes. Now this classical view is strongly challenged to favor an alternative view where the eukaryotic domain would have emerged from inside the archaeal domain. So we can, this is called the two-domain view of the tree of life. There is this big split between bacteria and archaea, and within archaea, the eukaryotic domain emerges. So that's possibly a better view than the classical one. The jury is still out. But we have done an experiment with using the alternative views of the tree of life, and this shows that our inferences are not sensitive to this aspect of the uncertainty about the tree of life. And lastly, what information in the molecules allow this non-parsimonious result? Well, it is the fact that a molecule, a protein sequence, or a ribosome RNA sequence, is made of several hundreds or several thousand sites, and each site evolves at its own rate. So some sites are slowly evolving, some sites are fast evolving, and this is where the modeling we use extracts information about able to reconstruct mesophilic ancestor to two thermophilic descendants. The fact is the slow evolving sites are closer to the ancestral state of the molecule, so they contain information about the temperature at which they contain more information about the temperature at which the ancestral organism lived, because those slow evolving sites today are still relatively close to the state they had very remotely in the past, whereas the fast evolving sites have been more transformed, and their present state informs us more about the shallower parts of the tree. And it is the fact that both kinds of sites coexist in the same molecule that allows our model to produce these non-parsimonious estimates that has surprised many of our readers. And to conclude, these are the fine colleagues with which I have been working on this topic. And thank you for your attention, and I hope we can exchange a few questions.